Hello, everyone, and welcome, dear students. This is Dr. Muzammil Yaqdali, Assistant Professor of Biochemistry. Today, we are diving into lecture number 3, the fatty acids, the chemistry, classification, and biochemical importance of fatty acids. So, fatty acids are the building blocks of most lipids, and understanding them is crucial for topics like membrane biology, nutrition, and metabolic diseases. If you find this lecture valuable, please like, share, and subscribe to our channel, and also press the bell icon for more updated lectures. So let's begin with the lecture, right? In this lecture, we will explore the structure, classification, physical properties, nomenclature, and biological importance of fatty acids. By the end of this lecture, you should be able to understand why some fats are solid, others liquid, and why essential fatty acids are called as essential. So let's start with the definition, right? A fatty acid is an organic acid found naturally in triglycerides. It is a monocarboxylic acid with the hydrocarbon chain ranging from 4 to 24 carbon atoms. The general formula is the RCOOH, where R is a long chain of carbon and the fatty acids are the building blocks of more complex lipids. So this illustration shows the fatty acid structure, right? The carboxylic group, the acid end, and the hydrophilic end. That is the main end. This carbon chain, that is the hydrophobic in nature and variable in length. And the methyl group, that is the terminal end. This amphipathic nature, both water-loving and water-bearing parts, is the key to how fats form membranes and micelles, right? So what about the general properties or the general features of the natural fatty acids? With few exceptions, natural fatty acids have even number of carbon atoms due to the synthesis from the two carbon units. They are unbranched chain or no side branches are present, right? Carboxylic group at one end and methyl group at the other. This uniformity allows predictable physiological or the physical and chemical behavior of fatty acids. So we classify fatty acids by the long chain length, right? The short chain means 2 to 6 carbon atoms. They are volatile fatty acids like the acetic or the butyric acid, right? The medium chain that contains 8 to 12 carbon atoms and found in coconut oil. And the long chain, 14 to 24 carbons, right? Most common in our diet and body. So rule number 1, rule for this length chain of the fatty acid is the longer the chain, the higher the melting point, as we discussed in the previous lecture. So most natural fatty acids have 16 to 18 carbon atoms, right? The carbons present in its structure, right? So what about the saturation? So saturation refers to the presence of or the absence of the double bonds. Saturated means no double bonds are present. Unsaturated means no one or more double bonds are present. So we can further classify the unsaturated fatty acids into monounsaturated fatty acids and the polyunsaturated fatty acids, right? Monounsaturated fatty acids mean there will be one double bond present in structure, and the poly means more than one. So two or more than two or more double bonds are present. So more double bonds are present means lower the melting point, or that fat or that fatty acid or the lipid will be liquid at the room temperature. So this is one of the hallmark feature of that particular fatty acid. So, double bond is the point of unsaturation, right? As the number of double bonds increases, the melting point decreases. So what about the saturated fats or its sources and examples? Saturated fats have all single bonds between carbons. They are fully saturated with hydrogen atoms. So mean there will be no double bond present in between the carbons, right? So what about the physical state? They are solid at temperature or the room temperature. The sources are butter, lard, coconut oil, palm oil, poultry skin, and the whole milk. So example are the acetic acid, butyric acid, palmitic acid, and stearic acid. So excess intake is linked to the cardiovascular disease. So what about the monounsaturated fatty acids? So they have one double bond. So what about the physical state? They are liquid at room temperature. The sources are the olive oil, canola oil, peanut oil, avocados, almonds, and cashews. The examples are the oleic acid. They have double bond at the carbon number 9. So they have 18 carbon structure. So these are heart healthy when used in moderation. So what about the polyunsaturated fatty acids? Polyunsaturated fatty acids contain two or more than two double bonds. They include the essential fatty acids. The examples are the omega, that is the linolenic acid, omega 6, that is the linoleic acid. So rich sources are the vegetable oils like corn, sunflower, safflower, and the cottonseed oil. So these are crucial for brain function, inflammation, control, and cell membrane structure, right? So what about the saturation and rancidity? So unsaturated fatty acids are more susceptible to rancidity because oxygen attacks double bonds, forming peroxides and free radicals. In ruminants, microbes in the rumen hydrogenate unsaturated fats to saturated fats. And the antioxidants like the vitamin E, vitamin C, carotenoids, and selenium help to prevent the oxidative damage, right? So what about the physical traits and membranes, mesols, and soaps? Because fatty acids are amphipathic, so they form lipid bilayer, form mesols during the fat digestion. So they also react with cations to form soaps. Sodium and potassium soaps, like they are water soluble. What about the calcium and magnesium soap? They are insoluble, poorly digested, and a challenge in ruminant nutrition, right? So what about the nomenclature of the fatty acids or the chain length? So fatty acids nomenclature follows a standard system. The chain length is written first. The C18 means 18 carbons. Most have even numbers, but fish oil contains odd chain fatty acids. So double bonds are indicated after a colon, right? So colon, so it means C18 colon 1 means 18 carbon and one double bond is present in its structure, right? So what about the examples, right? So nomenclature example like the stearic, oleic, linoleic, and linolenic acid. So let's see the examples. C18. Okay. So C18 colon 0 means stearic acid, that is fully saturated. C18 colon 1 means oleic acid, that is monounsaturated fatty acid. C18 colon 2 means linoleic acid, means polyunsaturated or the omega 6. What about the C18 colon 3 means linolenic acid, that is polyunsaturated or the omega 3. The number after colon is a double bond. The location matters biologically. So what about the fatty acid nomenclature omega system? The omega system numbers double bonds from the methyl end, the tail end. So omega 3, the first double bond at the carbon 3 from the methyl end. And the omega 6, that is means the N6, first double bond at the carbon number 6. And what about the omega-9? Means N9, first double bond at the carbon number 9. Animals can make omega-9 but cannot make omega-3 or omega-6, as they are essential in nature. So what about the isomerism in fatty acids, cis or trans? Double bonds create geometric isomers. Cis form means hydrogens on the same side, causes a bend in the chain, occurs naturally. And what about the trans form? That hydrogens on the opposite side, straight chain, rare in nature, but formed during industrial hydrogenation and the rumen bacteria. Trans fats are associated with the heart disease. So ruminant fats naturally contain some trans fats. So what about the melting temperature or the melting point, the chain length and saturation? Melting point is a key physical property. Effect on chain length or the effect of chain length means longer the chain, more will be the melting, the higher will be the melting point. So what about the example, right? C12:0 melts at the 44 degrees Celsius, while the C20:0 at the 77 degrees Celsius. So effect of unsaturation means more double bonds are present in that structure, longer the lower will be the melting point. So C18, the stearic acid, melts at 72 degrees Celsius, while the C18:3 at the 11 degrees Celsius, or the minus 11 degrees Celsius. This explains why plant oils are liquid and animal fats are solid in nature. So let's talk about the essential fatty acids. The definition: essential fatty acids cannot be synthesized by the body and must come from the diet. So examples are the linoleic acid, that is omega-6, and the linolenic acid, that is omega-3. And the third one is the arachidonic acid, is conditionally essential, can be made from linoleic and linolenic acid in the cats who require it directly. So what about the function of essential fatty acids? Essential fatty acids are vital for the cell membrane phospholipids, precursor for the eicosanoids, like the prostaglandin, thromboxane, leukotrienes, regulators of the smooth muscle contraction, platelet aggregation, and inflammation. Without them, our body cannot mount proper inflammatory and immune responses. So what about the ruminants and essential fatty acids? Here is a fascinating problem. In ruminants, cows and sheep, dietary polyunsaturated fatty acids are hydrogenated by rumen microbes into saturated fatty acids. So how do ruminants get essential fatty acids? So they use two strategies: rumen protective lipids, that is bypass fats, and the second one is the microbial synthesis. So microbes produce their own fatty acids for membranes, which the host animal absorb. This is a unique adaptation. So what about the problem with the deficiency of the essential fatty acids? Deficiency signs include growth retardation, reproductive failure, skin lesions, kidney and liver disorders. Fortunately, deficiencies are rare in humans due to the widespread vegetable oils and fish and diets, right? So fatty acids are everywhere in biochemistry, from cell membrane to hormones to heart disease. So master this and lipids will become much easier. If this lecture helped you, please like, share, and subscribe to our channel. Hit the notification bell or the bell icon so you never miss the upcoming lecture. This is Dr. Muzammil Yaqati signing off. See you in the next lecture. Thank you very much.